Friends, today we are going to study about nucleic acid part 1. In that, we'll study about its introduction, components of nucleic acids, type, structure and tautomeric forms of bases, bonds and its stability. What is the basic unit of nucleic acid? It is nucleotides. So nucleotides are the building blocks of nucleic acids. Now these nucleotides perform varieties of function. The first function, of course, we all know that it forms nucleic acid. Apart from that, other functions include energy currency. For example, ATP, GTP. These are what? These are nucleotides that function as an energy currency of the cell. In signal transduction pathways, for example, cyclic AMP function as a secondary messenger in various signal transduction pathways. And these nucleotides are also function as an important metabolic intermediates. Now, in our body, the nucleic acids, that is, there are two types of nucleic acids found in our body. One is DNA, that is deoxyribonucleic acid, RNA, that is ribonucleic acid. So these are the nucleic acid found in our body. Among these two, DNA function mostly as genetic material, but there are organisms where RNA can act as a genetic material, and mainly these are viruses where RNA acts as a genetic material. For example, as you all know, COVID virus, HIV, these are the important viruses that affects human containing RNA as a genetic material. Apart from these viruses, all other organisms, including bacteria, higher organism, plants, animals, all contain DNA as a genetic material. Now, this DNA, because it is a genetic material, it can be transformed from one generation to another generation. So DNA, because it is a genetic material, DNA can be transferred from one generation to the next generation. And in a given organism, from this DNA, RNA is formed, and from this RNA, proteins are made. So proteins are the end product of this pathway, and this pathway is known as central dogma. The central dogma, it is the pathway by which DNA is transcribed into RNA and RNA is translated into protein. So this pathway is known as central dogma. Now what is gene? Say for example, this whole length is a DNA. Now in this whole length, maybe this portion is responsible for the formation of a functional protein. So this particular portion will be considered as one gene. So in DNA, genes are those part of or the segment of DNA that is responsible for the formation of protein. That is considered as one gene. An organism contains multiple genes responsible for formation of multiple proteins. Now DNA, as I said just now, it is a deoxyribonucleic acid and RNA is ribonucleic acid. Now what is the difference between deoxyribo and the ribonucleic acid? We'll see in the subsequent slides. RNA is mainly categorized into three categories. rRNA, that is ribosomal RNA, mRNA, that is messenger RNA, and tRNA, that is transfer RNA. So these three are the major varieties of RNA found in a cell. But apart from these RNAs, there are multiple other RNAs found. For example, miRNA, snRNA, gRNA. So we'll study about these RNAs as well. So for now, RNA is mainly categorized into three classes, that is rRNA, ribosomal RNA, which is mainly function for the formation of ribosome. mRNA, it is messenger RNA. So in this pathway of central dogma from DNA to RNA and from RNA to protein, this RNA is mRNA. So mRNA carries the codons from DNA to form the protein. That is why it is known as messenger RNA. It acts as a messenger that carries the message stored in DNA to form a protein. What is transfer RNA? During the formation of a protein, the mRNA contains the codons. Like this, mRNA contains codon. Now, according to this codon, for example, AUG, this codon codes for amino acid methionine. Proteins are made up of amino acids. So that means to make a complete protein, we have to link the amino acids. So, for example, AUG code for amino acid methionine. Now, this codon is present in mRNA. mRNA is giving the message that in this particular protein, now we have to add methionine. But who will bring this amino acid methionine? That will be brought by tRNA. So hence it is known as transfer RNA because it is transferring the amino acid according to the codons present in mRNA. Now nucleotides. First, what are the components of nucleotides? So nucleotides have three components: nitrogenous base, pentose sugar, and phosphate. Now, if we remove this phosphate, so these two, that is base plus sugar, is a nucleoside. In nucleoside, when phosphate get attached, they form nucleotides. So what are nucleoside? Nucleoside is a sugar plus base. What is nucleotide? That is nucleoside plus phosphate. That means the combination of all the three. Now bases are flat heterocyclic ring consisting of carbon and nitrogen atom. Hence, these are known as nitrogenous base because it consists of carbon and nitrogen atoms. Now, bases are of two types, purines and pyrimidines. Purines are again divided into two types, adenine and guanine. Pyrimidines are of two types, cytosine and thymine in case of DNA. But if we see in RNA, then we'll have cytosine and uracil. Now this is the structure of purine. Purine is a doubled ring structure. From this nitrogen, it is numbered as nitrogen number 1 and then we move from the left hand side as 2, 3, 4, 5 and 6. Similarly, this nitrogen is 7 and then we move from the right hand side, we'll get 7, 8 and 9. Now two types of purines are adenine. Now this position is 6th position. So adenine can be called as 6-aminopurine. Similarly, for guanine, this position is 6th position. This position is 2nd position. So guanine can also be known as 2-amino-6-oxypurine. Now similarly, pyrimidine. This nitrogen is numbered as nitrogen 1. Then we can move from right hand side and it is numbered subsequently. Now two types of pyrimidines are cytosine and thymine. This position, 4th position. This position, 2nd position. So cytosine can be known as 2-oxy-4-amino-pyrimidine. Similarly for thymine, this position, 2nd position. This position, 4th position. This position, 5th position. So thymine can be named as 2,4-dioxy-5-methyl-pyrimidine. Now one more pyrimidine is there, that is uracil. Uracil is same as cytosine, only in place of this 4th amino group, there will be another oxy group. So uracil is same like cytosine, only the difference is here it will be one more oxy group in place of NH2 or amino group. So uracil then can be named as 2,4-dioxypyrimidine. Now pentose sugar. In case of DNA or RNA, the sugar is a ribose sugar. This is the structure, a ribose sugar, which is a 5-carbon sugar. The carbon number 1 is linked with a base, either purine or pyrimidine. The 5th carbon is linked with phosphate. And this form of structure is known as furan ring, furanose structure. Okay, with C1, it is joined with a base, that is purine or pyrimidine. With C5, it is joined with a phosphate group. If this is carbon number 1, this is carbon number 2. This is the structure of RNA. Now when we say DNA, that is deoxyribonucleic acid. That means in the 2nd position, oxygen is not present. That is deoxy. Oxygen is absent from the 2nd position in case of DNA. So this is of pentose sugar. Now, if we see this base, the base can be purine, which is a double ring structure, or pyrimidine, which is a single ring structure. In case of purine, this C1 is linked with the 9th nitrogen, N9. If it is a pyrimidine, then in that case, it will be linked with the 1st nitrogen, N1. So, if the bond is between sugar and a purine, then the sugar is C1 and the purine is N9. These two are bonded. If the bond is between sugar and pyrimidine, in that case, for sugar the position is same, that is C1. In case of pyrimidine, it is bonded with the nitrogen at position 1. So for sugar and purine, the bond is C1N9. For sugar and pyrimidine, the bond is C1 and N1. And this bond is known as glycosidic bond because the bond is connecting link between sugar and base. Hence, the bond is called glycosidic bond. Now with the C5 position, that means sugar and phosphate. This phosphate is attached to the sugar at C5 position, and this bond is known as phosphomonoester bond. Phospho comes from phosphate, mono means single, ester sugar. So phosphomonoester bond. Phosphate is attached with single sugar. Hence the bond is called phosphate monoester bond. Now here you can see this is the structure of deoxyribose because in the second position, as you can see, O is missing. Now with the C1 position, it is joined with the base. This base is adenine, that is purine. That's why it is linked with the N9. They'll form a bond with the release of water molecule. Similarly, this is C5 position of sugar is attached with phosphate. With the release of water, it will form the bond which is known as phosphomonoester bond. So that's how with the release of water molecules, C1 of the glucose is linked via glycosidic bond to a purine or pyrimidine, and C5 of the glucose is linked via phosphomonoester bond to a phosphoric acid or phosphate. Now this is the list of the nomenclature, how we pronounce, how we call. If the base is adenine, the nucleoside, that means when the phosphate is not attached, we'll call it as adenosine for RNA. If it is a DNA, we will call it as deoxyadenosine. If phosphate is present and it is a complete nucleotide, we'll call it as adenylate or deoxyadenylate. Similarly, for guanine, in case of nucleoside, we will call it as guanosine and deoxyguanosine. If it is a nucleotide, we will call it as a guanylate and deoxyguanylate. Similarly, for cytosine, it is cytidine and deoxycytidine for nucleoside. And for nucleotide, it is citidylate and deoxycitidylate. So, similarly, thymidine or deoxythymidine, thymidylate or deoxythymidylate. For uracil, it is uridine in case of nucleoside and uridylate in case of nucleotide. So this is the nomenclature for the bases with respect to nucleoside and nucleotides. Now the phosphodiester bonds, they link successive nucleotides in nucleic acid. So now this bond is known as phosphodiester bond. Why so? What is phosphomonoester bond? As we said that the fifth position of the sugar molecule is attached with phosphate. Hence it is phosphomonoester bond. That means single sugar is attached. But if we see the bond is between two sugar. This is one sugar and this is one sugar. How the bond is? C3 of one sugar, then phosphate, and then C5 of another sugar. That means phosphate is bonded with two sugar molecules. Hence, the name is phosphodiester bond. So, for nucleic acid, whether it is a DNA or it is a RNA, the backbone consists of this phosphate group and the ribose residue. So, the phosphodiester bond, that is the phosphate and the sugar, they constitute the backbone of the nucleic acid. And this backbone is hydrophilic in nature. The phosphate is also hydrophilic in nature. The sugar is also hydrophilic in nature. So the backbone is hydrophilic in nature. But if we see these bases are hydrophobic in nature. The backbone of DNA or RNA is hydrophilic in nature, but the bases are hydrophobic in nature. So the phosphoryl group between two nucleotides has one sugar esterified to it at 3' position and second sugar esterified to it at 5' or 5' position. So this creates an inherent polarity. Now the covalent backbone can undergo hydrolysis under alkaline condition. Now you can see alkaline condition means more of OH. So when there are more of OH in the medium, this OH will attack this hydrogen. because of which what will happen, this O will come and attack this phosphate. If this O attack this phosphate, then from here the bond will break. So what is happening? This is the shortened RNA because this bond has been broken and the link is between second position or 2' position and the 3' position. So like this 2' 3' cyclic monophosphate derivative will form in an alkaline condition. So when there is more of OH minus in the surrounding solvent, it will attack the 2' position of the pentose sugar, forming 2' 3' cyclic monophosphate derivative. Now DNA is resistant from this hydrolysis. Why so? Because DNA lack oxygen at 2' position. So because of this lack of oxygen, DNA is resistant from this hydrolysis, whereas RNA not. Now the properties of nucleotide bases that affects the 3D structure of nucleic acid. First of all, each of the bases exists in two alternative tautomeric states. Now here it is given cytosine. This red arrow is for hydrogen donor and the green arrow is for hydrogen acceptor. Now in the cytosine you can see this is the amino form. So this hydrogen will act as a donor for the bond formation. Whereas here nitrogen and these two positions, oxygen and nitrogen, they will act as a hydrogen acceptor. So this is the amino form. NH2 is there, no? So this is amino form. If H is missing from here, then it will become imino form. So in case of imino, this donor will become acceptor. Similarly for oxygen, oxygen is a acceptor, whereas two aminos, these are donors of hydrogen. This is the keto form. In case of enol, one hydrogen will get attached with this oxygen. So now it became from acceptor, it became donor. So in nucleic acid, the amino form and the keto forms are common. Rarely it can be in imino or enol form. So in case of nucleic acid, if you see, the amino forms are the hydrogen donor and the keto forms of oxygen are the hydrogen acceptor. In case of nitrogen, amino form is common and in case of oxygen, keto form is common. Here in cytosine, if we see this oxygen and this nitrogen acts as an acceptor. Now the double helix is stabilized by base pairing. Base pairing means in case of nucleic acid, A is paired with T and G is paired with C. Adenine is paired with thymine, guanine is paired with cytosine. So if we talk about the single length of DNA or RNA, then the bond we will see will be the phosphodiester bond. When this single length of DNA is in double helix form, then the bond between the two helix will be the hydrogen bond. So in one strand, if A is present, it will bind with T with the complementary strand. Number of hydrogen bonds? 2. If G is present in one strand, it will bond with C in the complementary strand with number of bonds? 3. So double helix is stabilized by base pairing. So what are the bonds that stabilizes the double helix? That is hydrogen bond, of course, the main stabilizing bond. Apart from that, because the double helix are very close with each other, the van der Waals interaction will come into play. Because the bases are hydrophobic in nature, so hydrophobic interactions will also come into play for the stabilization of the double helix. And this hydrophobic interaction is the result of base stacking. If this is the single strand of DNA, then the backbone will consist of sugar and then phosphate, then sugar, then phosphate. In this strand also, sugar, then phosphate, then sugar, then phosphate. The base will be present like this. So among them, there will be hydrogen bond. Because they are close enough, so van der Waals interaction will also come into play. And because these are hydrophobic bases, so the hydrophobic interaction will come into play. So this is because of base stacking, because the bases are stacked with each other. So here you can see the first one is the bond between G and C, 3 bonds, 1, 2 and 3. The positions are given, that is 6, 4, 1, 3, 2 and 2. Between these positions, the bonds are formed. Similarly, in case of A and T, 2 bonds are there. The positions are 6, 4, 1 and 3. So these are the positions where hydrogen bonds are formed. So this is the structure of a double helix where both the strands are joined together with the help of a hydrogen bond between the bases, and this creates a polarity. As you can see, the 3' position and the 5' position. So this creates a polarity because here 3' position is free. With this 3' position, no phosphates are attached. In the same strand, if we see below, this is the 5' position. That means with the 5' position, no sugar is attached. 3' OH and 5' phosphate groups. So this creates a polarity of 3' and 5' end. So now one question from my end for you all, that is, which positions in a purine ring of a purine nucleotide in DNA have the potential to form hydrogen bonds but are not involved in Watson-Crick base pairing? Forget about this Watson-Crick word. We'll study this when we'll study the structure of nucleic acid. For now, you have to answer that in case of purine, you have to identify the position that are capable of forming the hydrogen bond. But in case of DNA, that position is not forming the base pair with the complementary strand. So do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.